We were talking about genotyping last time, and we talked about you can genotype individuals. You just get a profile of what the alleles are that they're carrying, right? So it's a way to get a survey, basically, of what's in the genome, but you're not doing actual single nucleotide sequencing, right? And we talked about two different types. We talked about microsatellite genotyping, where you amplify these various fragment length regions and get a profile of what people's genome looks like. We also talked about restriction in fragments, right? Just taking the entire genome and just fragmenting it with restriction enzymes and running those out and using that as a means to genotype. I just want to finish that up and talk about the third way to genotype. And this introduces a method that we're going to see a lot, actually, in the next several lectures and for the rest of the semester, is this idea of blotting. So this is called a southern blot. And what it is it's a way to actually genotype, or you can do actually lots of different things with southern blotting, but we'll talk about how to use southern blotting for genotyping. What a southern blot is, well, it was developed by this guy named Edwin Southern. So it's named after him, right? So it's a blotting method named after this guy. What Southern did is he took, and in this case, we're using an example of paternity testing. Okay, we're looking at what the genotypes of a mother, father, and baby are, right? We're doing a paternity test. In southern blotting, what you do is you take DNA from the individuals you're looking at. This could be separate individuals, like in this case, or it could be different tissue types, it could be different developmental stages, whatever. You just get DNA from some source that we're wanting to compare, okay? You isolate all the DNA, and then you run all the DNA out on a gel. You'll probably fragment this first, so you probably do a restriction fragment. And you run out all these various sized pieces on the gel. Now in southern blotting, we don't actually look at all the bases, all the fragments on the gel. We are doing a selectively probing for the things that we want to look at, okay? So you've got all of this DNA out on the gel. You transfer all of those fragments onto a piece of nitrocellulose. This is what makes it a blot. So we're taking all those DNA fragments that used to be in our gel, and we've fragmented them and separated them out by size, and you just transfer them to a piece of nitrocellulose film, just a very thin film. So all of these streaks are all of the DNA that is in that sample. Then what we do is you probe for the pieces of DNA that you're interested in. And the way you do this is you make an antisense, you make a reverse complement sequence probe to the gene, the piece that you're interested in looking at. Okay, so you soak your blot now. So instead of being a gel now, we call it a blot because it's on this film. And you put it in your probe solution. And wherever the probe finds complementary sequence, your probe will stick. And then if we've got it labeled some way, the traditional way was to radioactively label it, but you could label it with some color change or some fluorescence or something like that. Then you just take a look at what actually shows up on your blot. So what we're looking at is specific allelic differences between these individuals in this paternity testing, right? So when we put in the probe is looking for a certain DNA sequence, and it's just going to stick wherever that is. Now here in the mother has these two small fragments representing pieces that were fragmented in her genome where this piece sticks. She has two different alleles, right? However the genome was fragmented, the location where this sequence was fragmented in two different sized pieces, corresponding to her two copies of the genome, right? So she's got a large band piece there, and she's got a small piece. The baby here has a small piece that she inherited from mom. That's where the probe is sticking here, is on that fragment that was specific to her mom's chromosome. And then a fragment that was specific from her father's. And then on the other here, the last lane is the father, right? These are the two alleles that he has. So the probe is just sticking to these random fragments, okay? And the size of the random fragments is what we're actually using to genotype. So the baby has got that allele from mom and that allele from dad, and so we've done a basic genotyping using southern blotting. So a southern blot is really just a generic term for running DNA out on a gel and probing it with an antisense piece of DNA. And you could probe this blot for multiple things, right? We made a probe that sticks selectively to that sequence. You could wash this though, all that probe would come off, and you could blot it and probe it again for a different sequence if you wanted. Make a different probe that hybridizes to a different sequence, and you could do this over and over again. This is a southern blot. Yeah? Yeah.
I am wondering if it's on the gel, and then is it actually all the DNA sequence or fragments that are transferred to the film?
Yeah, everything that was in the gel has been transferred onto the blot. Yeah, to the nitrocellulose.
And
So that streak is just like all the fragments that were in the sample, right? They're all just stacking on top of each other.
and for the last step, how I know you said like we can repeat and do different probing,
Mm-hmm.
but is it using the same film?
Yeah, you would use the same film again. So you could repeat this again because you've transferred all the DNA from the gel onto this nitrocellulose, and you've permanently covalently attached the DNA to the film, or to the nitrocellulose. Right? It's stuck there permanently. The only reason your probe is sticking to these places is because it's just base pairing. Right? It's matching up with this complementary sequence. So you can easily just wash this in some high salt, and you can break the base pairing apart, and the probe will come off. But all of the DNA is covalently bound to it. It'll stay permanently. So you can probe it over and over again. Just wash the probe off, put a different probe on. But all that DNA is actually permanently covalently bonded. There's all kinds of different blotting. This is southern blotting. Northern blotting is, instead of using DNA out on your gel and transferring it to the nitrocellulose, instead you would take mRNA from your samples. So you could compare what genes are expressed. I could take liver tissue and kidney tissue and brain tissue, extract out all the mRNA and run that out on a gel, and then I could probe for the presence of a certain gene being expressed. Right? If I say, like, I wonder if gene X is expressed in kidney or liver or brain, well, I could make a probe to gene X. I could run out all of the mRNA from those tissues, and then my northern blot would show me if the probe sticks to liver tissue sample, then that gene got expressed at the mRNA level in the liver tissue. Right? So you can blot this for multiple things. So that's a northern blot, is when you use RNA as your template. Western blot is when you run out protein on a gel and then probe it with antibodies that stick to protein sample. So you can, this is two ways to look at gene expression, right? At the mRNA level or at the actual protein level. Does it get made into protein in the tissue? So, and then there's all kinds of clever variations on this as well. Don't worry about all these, but there's Eastern blotting and Near Eastern and Far Eastern and Far Western, and it's just basically changing what we ran out on the gel and how we're probing it. So anytime you change what you're looking for and what the probe is, they just named it something directional. Now, there was no Mr. Northern, there was no Mr. Western. These are all just jokes made on the original guy that developed it. So, all right, so I just want you to understand that you can probe a blot in order to do genotyping, right? Okay, we're gonna segue now and start talking about transgenics. Now, we've actually already talked about transgenics a little bit. If you make a recombinant plasmid, right, if you take a plasmid and you put your piece of DNA in it and you get that taken up by a bacteria, if you transform bacteria with something other than bacterial DNA, you have made a transgenic organism, right? But most of the time, that's not what we think about when we think about transgenic organisms. When we think about transgenic organisms, we think about making organisms express genes that they normally don't express, right? That bacteria that's holding that plasmid, all it's doing is holding a sequence of DNA so you can conveniently make up lots of copies of it. But it's not making, we cloned the piece of the senseless gene, and we've got bacteria that currently have that inside them right now. You've made transgenic bacteria, but they're not expressing the senseless gene. They're not making fly proteins. They're just holding that piece of DNA for you. So I'm going to talk about what we call expression constructs or expression vectors. These are ways that we're going to clone in pieces of DNA and get the organism that we put this DNA into to actually express these genes. They will actually make mRNA off of the gene that you've cloned in. It will turn that mRNA into protein, and then you'll have actually conferred a new trait onto the transgenic organism. So we need to talk about how you actually do that, and so we'll just talk a little bit about what changes a normal vector into an expression vector that will actually make your mRNA. Okay? So here's an example of an expression vector. It's got an origin of replication. It's got an ampicillin resistance gene. So this is a expression vector that's based on a bacterial plasmid. This would grow in bacteria, right? And then we have put luciferase. In green here is the coding sequence for a gene called luciferase. Luciferase is the protein that fireflies make in their abdomen that glows, right? It's a protein that actually emits light. So it takes energy from the cell, it metabolizes that energy, and gives off wavelengths of light. It actually glows in the dark, right? So what I'm going to do is try and get a bacteria that will actually make this luciferase. There's a couple of things that I have to do, right? You can't just grab the luciferase gene out of fireflies and stick it into a bacterial plasmid and expect it to just be made into mRNA and made into protein. We're going to have to manipulate this a little bit and trick the bacteria into thinking that this is a gene that it should express, right? So an expression vector has a couple of things in it. I'll go for the bottom up, actually. It has to have 3' and 5' UTR regions that a bacteria thinks are the appropriate 5' and 3' UTR regions, right? The firefly might have the UTR act a certain way, but if the bacteria doesn't recognize that, then it might not put the poly A tail on. It might not put the 5' cap on. It might not keep that around, right? So I have to put appropriate 3' and 5' UTRs for whatever host I'm trying to get this to be made in. The other is you need to have an RNA polymerase binding site. Right? What we want is the bacteria to make this into mRNA. So we have to put in the plasmid the sequence where the bacteria's RNA polymerase wants to sit down and would actually make this into mRNA. So incorporated into that expression vector, you've got to get basal promoters, right? If it's bacteria, you have to put a bacterial promoter that it will recognize. If you're trying to make a transgenic corn plant, you're going to need to put a promoter that a corn is going to recognize so that it will actually make your protein, your transgenic protein, okay? Now, there's kind of two ways that you can work this. Now, you have to have a place for RNA polymerase to sit down at least, right? If it's going to get made into mRNA, the bacteria actually has to sit its RNA polymerase down. But then you also want it to get turned on, right? And so having just an RNA polymerase binding site is not enough. You have to have a place where transcription factors actually say, yeah, this is the kind of gene that we want to turn on right now, okay? So a basal promoter is got to have an RNA polymerase binding site, but then you probably have to have an enhancer region that transcription factors will sit down on and say, yeah, I'm actually going to recruit RNA polymerase to come sit down on here and make this protein, or make this mRNA in appreciable amounts. So a promoter that regulates the expression. Now you can manipulate this multiple ways. You could, option number one, is you could put a promoter there that basically just tells the bacteria, this is a gene that you really want expressed all the time. You know, bacteria are expressing, metabolism genes in pretty high amounts, right? Bacteria like to feed on sugar, they do glycolysis. So if you took the promoter region of one of the glycolysis enzymes and stuck it in here, in this promoter region, then when I got this plasmid into the bacteria, it thinks that what it's doing is expressing this metabolism gene, because you've hooked up the promoter for the metabolism gene there. And so it will recognize it and say, oh, this is a gene I should express. But then the mRNA that it actually makes, you've tricked it into making the mRNA for Lucifer Ace. Right? Oftentimes, if you want this always on, you usually put a viral promoter. Viruses are really good at getting their DNA into host cells and tricking the host cell into making lots of copies of their own proteins. So if you want to always turn this gene on, then you usually put a viral promoter in there, a virus that would normally infect this kind of cell. Right? So if you're trying to do it in a bacteria, you would use like a bacterial phage promoter that would turn this gene on at high levels. If you were trying to do it in corn, you would use a virus that infects corn, and there are corn viruses. You take a corn virus promoter, stick it in front of this, and then when you get it into corn, corn would think, oh, well, it's commonly tricked, so the corn actually would rather not make corn virus, but corn virus is good at tricking corn into making its proteins, right? So you could trick the corn into making your Lucifer Ace gene. Yeah?
Would the addition of like the promoter in this vector like affect the regulation of like the actual gene that the promoter?
No, you can pretty much think that it's just an addition to what it would normally do. Yeah. So if you add a transgenic thing like this, it's not going to make the gene that normally uses this promoter to get down-regulated or something like that. Yeah. It's usually over and above. All right, so that's option one. You can just get whatever gene you want to be transgenic always turned on, no matter what. If the cell has this transgenic plasmid, then it's just always going to make luciferase. Option number two is you could stick a promoter in that is specific to a gene that you're interested in studying in that organism. So for instance, if I want to know when a certain gene gets turned on in brain cells, I could take the promoter of that gene that I'm interested in, and I could stick it into an expression vector, right? So I would take the promoter of gene, whatever gene you're interested in, this neural gene. I would stick that promoter in here, and then I would get this to actually be in the brain cells that I'm curious about. When those brain cells actually express my gene, they will also then express the luciferase. Whenever that promoter gets used, so whenever a cell tries to turn on the gene I'm interested in, it's going to turn on that gene, and it will also turn on the gene for luciferase. So every cell that's expressing the gene I'm interested in also starts glowing. Right? So if I'm looking at brain tissue under the microscope, you could actually see glowing cells indicating that the gene that I was curious about is actually being expressed in those cells. This is called a reporter construct. So it reports back to you when that promoter is getting activated, when that gene is getting expressed, because in addition to that gene, it's also going to turn on the luciferase. Questions about an expression vector? This is going to be the end of all new content. I'm just going to show you pretty pictures of expression vectors after this. So let's get clear on what an expression vector is and how it works, and then I'll just show you cool examples of how you've manipulated this. Mark, yeah. Restate what I've talked about for the last 20 minutes. No, sorry, go ahead.
Could you restate option number two? I understand option number one.
Okay. Option number two is, we're talking about the promoter that you put here, right? Option number one, you always put a promoter that gets turned on by the host cell. Option two is you put a promoter for a gene that you're interested in its expression, right? So it's just going to get turned on specific cell types. You know, you put a promoter for a gene that only gets turned on in neurons, right? And I want to see when do those neurons actually turn this gene on. Well, I would take that promoter, stick it in front of luciferase, and get this whole thing to be taken up by the brain cells that I'm curious about. Then whenever those brain cells want to turn on the gene you're interested in, they send transcription factors and RNA polymerase to sit down on that promoter and make that gene. But in addition, you've got your reporter in there. So it sits down on the promoter for the gene that it actually wants to express, and it sits down on the promoter here and turns luciferase on. So anytime your gene gets expressed, luciferase gets expressed too. And so the luciferase is reporting back to you when that gene is expressed. Right. Yeah, we'll do Olivia and then.
Okay, for the option number 2, the reporter construct,
Mhm.
the first option, the constitutive, right? If that one's
Yeah, we call that constitutive, yeah,
constitutive.
or always on, yeah.
Okay, so if that one's usually a viral promoter, then
Mhm.
the reporter construct, is that usually bacterial or?
No, I mean, if you wanted to do it in corn, you could use a corn virus, right?
So can
If you want to do it in human cells, you could use a cell that often, so cytomegalovirus, CMV, that's a human virus that infects human cells. So if you've got cells growing in tissue culture and you want them to express this, you would put CMV on it. And then when this gets into the cell, the promoter gets recognized by the cell and turns on the luciferase gene,
okay, so option,
mimicking the way that the virus would trick the cell into getting its genes turned on.
so option number 2 is basically variable.
Yeah, I mean, all of these are dependent upon what kind of cell are you getting them into, right? If it's in a human cell, then you've got to put 5' and 3' UTRs that a human cell would recognize. And you've got to have an RNA polymerase binding site that human cells recognize. And you have to have an enhancer region, whatever that promoter is, for a promoter that the human would recognize, human cells, yeah. This is all very, very host specific, right? Some things can kind of go crossways, and some organisms will recognize other organisms' promoters, but usually you got to be pretty specific to the cell type that you're trying to go into.
I think you sort of mentioned, that was my question. My question was, do we put these vectors into like a living animal, like a mouse, or?
Yeah, I'll show you examples of
Oh.
putting these in the mouse. It's pretty cool, actually.
Because you mentioned that we grow in labs, like
Mhm.
we put it in cells and then let them grow and then put it in a mouse, or?
Yeah, you can do it both ways. So I'll show you an example of both, actually.
Okay.
Yeah. I'm going to give you a C-squirt example first. Okay. This is an example of C-squirts, and instead of being luciferase, the reporter is the LAC-Z gene. It's the same gene that we're using in lab to tell us whether there was an insert or not. It's the gene that cleaves this lactose, right? So our reporter is the enzyme beta-galactosidase. It will cleave that XGAL molecule, and so if the cell is expressing this gene that we're interested in, it's going to be expressing beta-galactosidase. So if we soak the tissue in XGAL, every cell will turn blue that's expressing our gene. Okay. So this is a construct that was made. It's got the promoter region of a gene called brachyury. So anytime this gene brachyury gets turned on, it's going to turn on the beta-galactosidase gene. And if we get that tissue in the presence of XGAL, all those cells will turn blue. So this is a little C-squirt tadpole. So that's the adult C-squirt. Here's the cute little tadpole. It's actually still an embryo. This would be before it actually hatches out. If you actually took that and made this a transgenic animal, so what the researchers did here is they took a fertilized egg, actually they took hundreds of fertilized eggs, soaked them with this reporter construct, the brachyury gene hooked up to the brachyury promoter hooked up to the beta-galactosidase gene. And they actually either injected that DNA into the single cell zygote, the fertilized egg, or they like zapped all of the cells and forced them to like suck up DNA from the outside. You can kind of like you transform bacteria, you can kind of get other cells to take up the DNA. So all of the cells of this embryo have the reporter construct, but only these cells in the notochord actually turn on the brachyury gene. When they turn on the brachyury gene, they turn on our expression vector they turn on the beta-galactosidase gene. You have to fix the tissue, so I have to kill the embryo, fix it, and then I soak it in XGAL, and then every one of these cells that was expressing that gene is now showing up as blue. So I'm looking at exactly which cells in the C-squirt were expressing brachyury because I can see the reporter was expressed at the same time. This is the quick and dirty method. This is pretty exciting, right? I'm looking at actual gene expression in the cells of the tissue. And you could do this at every developmental stage, right? So you could grow up some of these transgenic embryos and stop them at an early stage and look to see, is brachyury expressed? You grow them up to a later stage, fix them, soak them in XGAL, see if brachyury is expressed. And so you can watch actual cells change in their expression pattern. A cell might not be expressing it at one developmental time point, but let it develop for 3 hours later, it might start turning on that gene. And you'll actually see that because the blue color will show up in those embryos then. So. This is an example of a Lucifer Ace reporter. So here's luciferase being expressed in the cells of the abdomen of the firefly, right? And so they're emitting photons of light. And so if it's dark outside and you've got a firefly, you can see it actually. Have you ever caught fireflies? You can crush them and smear them on your pants, and then and you've got luciferase and you're making yourself fluorescent. It's kind of morbid, but it's kind of fun too. In this example, what they've done is they've hooked up a luciferase reporter to a gene that gets turned on in a certain type of cancer. Okay, so in certain cancers, they upregulate certain genes. And one of the things they upregulate is the genes for metabolizing, right? They're actively growing and dividing cells, so they turn on in really high amounts metabolism genes and cell division genes. So I'm not going to go into what gene this is, but it's a gene that's normally turned on in gliomas. This is a cancer of the glial cells in your brain. Okay, so what they've been doing is growing these glial cells from a mouse in a petri dish, and they made them transgenic for this one gene. So whenever the glioma turns on this gene, it starts glowing. So the cells in the culture are actually growing. What they did is they took some of those and actually implanted them into a normal mouse. Okay, so they're growing these cancer cells. Now these cancer cells are growing in the petri dish, and they're also glowing with luciferase. Well, they took some of the cells and injected them in an adult, healthy rat that didn't have cancer, or mouse here. So they just subcutaneously just went in and injected some of these cancer cells. And so this is a live mouse. Right? He's not dead. You just put him to sleep temporarily. And what they're doing is actually taking a picture of him under infrared light, or an infrared detector. And so what you're seeing here is a picture of the rat under regular light, and then a picture overlaid on top of it of a detector that's detecting just the tiny photons of light that are being emitted from the cancer cells that are underneath the skin of this rat. So the little blue indicates to you how many cancer cells did you actually inject into that mouse. Then you let the mouse go for a couple of months, and you're watching how does the cancer progress, right? So then I can take that same mouse, a couple of months later, put him on the imaging machine, and now all of those cancer cells have metastasized, they're growing, they're dividing, and you can actually watch the cancer cells grow, but I haven't had to do anything to this mouse, right? All I did was put him on an infrared detector and just look to see the fluorescing, because some of those wavelengths of light that are being emitted from those cancer cells are making it through the skin and reporting back on that detector. So you can actually keep track of actual cell division and the progress of a disease because I've got a reporter gene for all of those diseased cells. So this is kind of a transgenic animal, not really, right? It's transgenic cancer cells that we just implanted just to watch the progression of the disease. So, yeah, people give mice cancer all the time. we're trying to cure the cancer, right? So, but. Another common reporter is green fluorescent protein. This is a protein that jellyfish actually make. Can you see this little jellyfish here? Jellyfish can absorb certain wavelengths of light and then they fluoresce them back. Jellyfish actually do really interesting communication by flashing different wavelengths of light at each other. But green fluorescent protein is a really common transgenic reporter gene. This is a worm, the little nematode worm that Dr. Cruzen works on. This is an adult worm. This is an adult worm that has green fluorescent protein attached to the promoter of a gene that gets turned on in all the neural cells. So every neuron, every neural cell in this worm is also fluorescing green fluorescent protein. Now this isn't Lucifer Ace. Lucifer Ace just emits light. Green fluorescent protein, you have to shine one wave of light at it, it absorbs it, and then it reflects it back to you. So this guy doesn't normally glow. He only glows if you shine certain wavelengths of light at him. But what we're looking at is now the entire neural network of a living worm, right? This worm is living. It's just expressing green fluorescent protein in every one of its neurons. You can also use this to make transgenic fish. These are actually called glowfish. This company has actually taken green fluorescent protein and its variants. So this is green fluorescent protein. There's red fluorescent protein, there's yellow fluorescent protein, there's cyan, or kind of a bluish fluorescent protein. And what they're doing here is they've got these different fluorescent proteins being expressed in the skin cells of these fish. So normally these fish, I don't have a regular, they're kind of just a dull kind of brownish tan color with some black spots on them. They've made these fish transgenic, and so this one's glowing green and this one's glowing red and yellow because they've got a transgenic gene that's being expressed in all of their skin cells. Right? Sea squirts. This is green fluorescent protein in the nervous system of the sea squirt. This is a normal wild type sea squirt, and this is its little brain cavity, and so you can see all the neurons are glowing. This is a little sensory organ. This is the mutant that I studied. So instead of having distinct little sensory organs, he doesn't know which cells should be neurons, so actually expressing green fluorescent protein in all these inappropriate cells. So it's a method by which you can detect abnormalities in development and things too, right? So I'm using this as a marker just to see what's going on in the nervous system of this guy, right? And he's kind of got a messed up brain too. It's got lots of problems. This is a transgenic bacteria artwork. So this is green fluorescent protein that they streaked out on a plate, and under fluorescent light, it's glowing green. So under normal light, this just looks like bacteria spread out, and you can't really see the picture, right? You might be able to kind of see what it is. But you put it under fluorescent light, and all these cells grow green, and here's red fluorescent protein, and blue, or this is cyan, right? Green and yellow and orange. A little purple up here too. So you can do artwork, but mostly it's used as a tool, right, to report back to you what genes are being expressed. So. Questions about reporter constructs? Yeah?
So on the study guide, it says we should know the types of reporter genes. How specific should it be? So for instance, should we just know green fluorescent protein?
You should know the three that I've talked about.
Do we know
LAC-Z. I mean,
for instance, LAC-Z is the bacterial gene that codes for and then
beta-galactosidase, yeah. So LAC-Z, if you give it X-gal, it turns blue. I mean, that's what you should know. Lucifer Ace just emits light on its own. Fluorescent proteins, you have to shine a wavelength of light at them, and then they fluoresce back to you, right? So if you just know those three types of reporters, It's good enough for now. Fluorescent proteins like these guys.
So refluorescence you shine with like that.
Yeah, so a fluorescing molecule doesn't actually shine, it doesn't just emit light on its own. You have to shine certain wavelengths of light at the protein. It absorbs those wavelengths, and then it emits photons of light at a different wavelength back at you. So that's fluorescing. You have to actually expose it to light. Luciferase just glows in the dark, right? You could take it into a dark room, you don't show it any wavelengths of light, and it just emits light at you, right? So that's luminescence. Fluorescence is when it actually absorbs one wavelength, so you have to shine it one wavelength, then it'll give you back a different one. Yeah?
What determines which type of reporter gene you use? It's just
Yeah,
application or the
yeah, just what you want to do. Yeah. I mean, if you want to do it in a living system, you want to keep the animal alive, then you need to do fluorescence or luciferase. Because if you're going to use LAC-Z, you got to soak it in this toxic X-gal, and you got to make the cells permeable to get the X-gal into the cells. So you have to fix that tissue. You have to kill the embryo, or fix the tissue, kill the cells, and then look at the expression. Fluorescence and luminescence are nice because you could just have a living cell, emitting photons of light. That's not going to kill the cell to be glowing, right?
Wouldn't it be that luciferase has a better application because either way it goes in dark as compared to the fluorescent proteins?
Yeah, the problem with the question is, wouldn't luciferase be a better option always? Luciferase, the wavelengths of light are very weak. And in fluorescence, well, number one, in fluorescence you can choose when it glows and when it doesn't, right? So you could take a picture of it without it glowing and then take a picture of it glowing. Luciferase, it's always there. Luciferase is fairly weak too, and so if you want really robust, strong detection, then fluorescence is the way. If you can't, for whatever reason, expose your cells to the right wavelengths, then you might need to go luminescence and use the luciferase. So it really just depends on whatever the constraints of your application and your experiment, right?
Also,
So don't worry about those. Just know what they are for now.
Luciferase is also cheaper, right? Because it's more common.
No, neither one of these is cheaper, really. I mean, people have got luciferase cloned into all kinds of vectors, and you could just order it from a company and they'll send you luciferase. They'll send you GFP. They'll send you any of these guys, right? So, I mean, in terms of like availability, and you're just going to pay for a vector, and whatever it's got in it, it's got in it. So, luciferase is probably a little bit cheaper on equipment-wise, right? Because you could just take your sample in the dark and look at it. If you're doing a fluorescence, then you have to have, fluorescent wavelengths and a fluorescent detector, and so. All right. Next time, after our exam, we'll actually talk about modifying these for actual individual cell types and such. But for right now, I just want you to get the idea of an expression vector.
Biola University offers a variety of biblically centered degree programs, ranging from business to ministry to the arts and sciences. Visit biola.edu to find out how Biola could make a difference in your life.